This IB biology revision video, Mr. Phillips, and this is for the respiration module. And just to remind you, this video does not contain all the content you need. It needs to be supported by a thorough revision yourself and work in lessons. So today we're going to look at all the stages of respiration, starting with C1.2 ATP production. You need to know the basic structure of ATP. You've probably already gone through this in the biomolecules module, but if we were to look at ATP, you need to be aware it's made of three main areas. So you have this area here, which is called adenine, and it's an organic base, so you'll recognize that from the DNA structure. That is attached to a 5-carbon sugar called ribose. So adenine and ribose make up something called adenosine. So adenine, ribose and 3 phosphate groups make up the structure of ATP. ATP is what we call the universal energy currency. So that means all living things use ATP. And using water, it can be hydrolyzed, so during a hydrolysis reaction, and broken down into ADP, adenosine diphosphate, plus water. It can be further broken down smaller and smaller, but we'll only really study about it being transferred into ADP and then back to ATP, which is the purpose of respiration. It cannot be transferred from cell to cell, so whichever cell it's made in is where it needs to be used, and it is catalyzed the breakdown of ATP using ATP hydrolase, which should make sense because it's a hydrolysis reaction. It has some essential properties that allow it to do its job for energy storage. So it stores and releases a manageable amount of energy only, and that reduces the amount of energy lost as heat waste. It is small and soluble, so it can move through the cytoplasm. It is easily broken down. It is quick to remake. It can also be used to make other molecules more reactive. So we're not going to talk about this in this video, but this is an essential part of the property. Actually, we will mention it slightly in glycolysis. It cannot leave the cell, and it is stable at a neutral pH. So then, what do we need to know about using ATP for energy? Well, there's a couple of key things that we need to know, key processes. So first of all, there is reactions that are endothermic that use energy, use ATP in the cell. So this is when we synthesize macromolecules, so using anabolic reactions. If you know what anabolic means, go back to the enzymes video. But anabolic is building up smaller molecules into bigger molecules. So things like DNA replication, making proteins from amino acids, all need energy. Anytime we're talking about active transport, we're using energy to pump ions against the concentration gradients, and therefore that is going to use energy to change the shape of the protein pump from one conformation to the other. And anytime movement is needed, either in cells or muscles, that will use energy as well. So there's a constant cycle going on with ATP. So basically ATP starts off, we use the ATP, breaking it down in a hydrolysis reaction to release the energy to make ADP and a phosphate. Then respiration does condensation reaction effectively to put it back to ATP. And that is being constantly cycled. Your body needs approximately 50 kilograms of ATP per day. You only ever have 5 grams in your body. So this cycle is consistently going on throughout your And again, just to remind you, it is a mixture of a hydrolysis reaction and condensation reaction. Respiration basically uses energy that is always released from carbon compounds. So whilst we're talking about glucose as the main one, fatty acids is also used in key parts of the body, such as the heart, and gas exchange therefore is vital, because as you should know, the basic reaction for respiration, or for aerobic respiration, is glucose C6H12O6 plus oxygen goes to carbon dioxide and water, and this releases energy in the form of heat, but it also makes ATP. When we're comparing aerobic to anaerobic respiration, then aerobic respiration, the overall equation is this one, which you should be familiar with. And again, you should know the symbol equation, which is on the previous slide. Anaerobic, there's two main forms. There's anaerobic in muscles during vigorous exercise, which would be therefore mammals and and that is converting glucose with no oxygen into lactic acid, and that produces less energy. And then in yeast and plants, I should say, there is glucose converted with no oxygen into alcohol and carbon dioxide, sometimes known as the ethanol fermentation process. And again, if you're comparing them, you should be able to identify the reactants. So we should be able to say that glucose and oxygen is the reactant for aerobic, just glucose for anaerobic. The combustion is incomplete. So think about chemistry. Incomplete combustion means there's not enough oxygen or no oxygen. Complete combustion means there is oxygen. The energy yield is a lot lower for anaerobic respiration. You only get 2 ATPs compared to a much higher number for aerobic. Products are obviously different, as you can see there, but notice carbon dioxide is produced in the anaerobic respiration of yeast and aerobic of all organisms. The location is important, so this happens in the cytoplasm for anaerobic, but in both the cytoplasm and the mitochondria for aerobic. And the stages, there are lots of stages of aerobic. There's only really the 2 stages, well, really 1 stage of anaerobic. They both require glucose, but then there are key differences. So continuing to compare the two, when you do anaerobic respiration as a human, as a mammal, we get oxygen debt. And that is because lactic acid, which can also be called lactate, okay, is a waste product that must be broken down because it lowers the pH in your cells. That would affect your enzymes over a long period of time, so it causes muscle fatigue and pain. This therefore requires extra oxygen. So oxygen debt, when you finish exercising, you breathe heavily to remove and break down that lactate and break down that lactate, which is a harmful product. So if we go on then to the first stage of respiration, which is glycolysis. There are basically 4 key reactions. We're talking about glycolysis in aerobic respiration, followed by the link reaction, followed by the Krebs cycle, and then oxidative phosphorylation. And let's have a look at how that process. Before we do that, you need to know what oxidation is. You should know the term oil rig. Oxidation is loss and reduction is gain. So when we're talking about oxidation reduction in respiration, we're really focusing on hydrogen ions, which we'll be talking about throughout this module. So glycolysis is the first stage of aerobic, and I said earlier, really the only main stage of anaerobic respiration. It's an ancient biochemical pathway which all cells use, and therefore we know it's evolved a long, long time ago. You only need to know an outline of it. The overall reaction is this: you take 1 molecule of glucose and you break it down into 2 molecules of pyruvate, plus other products, which we'll talk about in a moment. Picture summarizes the overall stage of glycolysis. So if we start off at the top, you have glucose, which is a 6-carbon compound. The first thing that has to happen, remember we said one of the jobs of ATP is activation of molecules to make them more reactive. Glucose is not very reactive. So we actually use 2 ATPs up in glucose to turn the glucose into phosphorylated glucose to make the molecule more reactive. So 2 ATPs are used, losing phosphate. That molecule of phosphorylated glucose breaks down then and splits into 2 molecules of triose phosphate, which is 3 carbon. And then there's a series of reactions which you don't need to know the full stages for because it's extremely complex in real life, where NAD, which is a coenzyme, is reduced. So what that means is it takes some hydrogen from the triose phosphate away, and that takes the final stage, which we'll talk about in a few minutes, oxidative phosphorylation. Also during this reaction, each molecule of triose phosphate is oxidized into pyruvate, and as it does this, 2 molecules of ATP are made and 2 molecules of ATP are made in this stage. So the overall products that you need to know is each glycolysis reaction makes 2 molecules of pyruvate, 2 molecules of reduced NAD, and 4 molecules of ATP. However, 4 molecules of ATP you have actually used up 2, so you only get a net gain of 2. And when we make ATP in this stage, when it goes from one substrate to the next, it's called substrate level phosphorylation. So then we've got molecules of ATP, we've got hydrogens, and we've got 2 molecules of pyruvate. So what happens to that pyruvate? Well, let's have a look in the link. And so first of all, the pyruvate has to enter the mitochondria, and you don't need to know this in detail, but it enters via a protein called pyruvate H plus symport, which allows the pyruvate and the hydrogen ions in the reduced NAD to go into the mitochondrial matrix. This uses energy, so it's a form of active transport, and it's one reason respiration is not 100% efficient. The first stage then is the link reaction. Now do remember as I'm talking about this, that actually we have 2 lots of pyruvate molecule here. So there really is 2 lots of pyruvate per glucose molecule. So what happens is the pyruvate comes in, and there is an enzyme called a decarboxylase enzyme which removes carbon dioxide, and that then diffuses out of the mitochondria into the cytoplasm, and then it's one of the waste products you excrete. NAD is further reduced, so again this coenzyme NAD takes hydrogens from the pyruvate, and this process is dehydrogenase. So this process of removing hydrogens and carbon dioxides creates acetate. Now acetate is not very useful in entering the next stage called the Krebs cycle. So a coenzyme called coenzyme A attaches to the acetate to make acetyl coenzyme A. That then takes the acetate into the Krebs cycle before the coenzyme gets recycled and reused again. So the coenzyme is like a bus taking the passenger acetate into the Krebs cycle. At this stage, we are making certain things. So you need to know for each molecule of glucose, don't forget there's 2 pyruvates. So we actually react that with 2 oxidized NADs and need 2 coenzyme A's to make 2 carbon dioxides, 2 reduced NADs, and 2 acetyl coenzyme A's. No ATP is made in the link reaction. The link reaction then we go into, so here you can see the acetyl coenzyme A drops the acetate in, the acetyl coenzyme A recycles itself, okay, sorry, the coenzyme A recycles itself, and the acetate attaches itself to a 4-carbon molecule called oxaloacetate to make a 6-carbon molecule called citrate. There is then a series of reactions where NAD is further reduced, so we're taking off hydrogens again, we're oxidizing the molecules, and carbon dioxide is removed to produce 2 carbon dioxides per cycle. Another coenzyme called FAD is reduced by adding hydrogens, and ATP is produced in substrate level phosphorylation. This cycle, for every glucose molecule, don't forget there are 2 pyruvates, so this cycle has to happen 2 times. In terms of what is actually produced, in the link reaction, 2 molecules of reduced NAD, which means they're carrying hydrogens, are reduced. In the Krebs cycle, this is per glucose molecule, I should say, because this means it's happening twice. The Krebs cycle happens 2 times every glucose molecule, so there are 6 molecules of reduced NAD. There is no reduced FAD carrying hydrogens in the link reaction, but 1 per cycle of the Krebs cycle, so overall 2 per glucose molecules. Carbon dioxide, 2 carbon dioxides are produced in the link reaction per glucose, 4 in the Krebs cycle, and ATP, there is 1 per cycle of the Krebs cycle. That goes around twice for every glucose, so 2 ATP molecules. You need to know the products of the Krebs cycle. We then look at what happens to these reduced NADs and reduced FADs that we have been talking about so much. This shows us the electron transport chain in an oxidative phosphorylation. So we've been talking about reduced NADs, which means they're effectively carrying hydrogen atoms. The hydrogen atoms, when they come to this final stage and to the cytochrome 1 here, is oxidized, releasing the hydrogens, and then the oxidized NAD go back to the previous stages to collect more hydrogens. Those, the electrons, just like in photosynthesis, are then carried through electron carriers. As they go through electron carriers, they lose energy, and that energy is used to pump hydrogens from the matrix of the mitochondria into the inner mitochondrial space. Now, when they get this, then builds up a proton gradient in terrible writing in the intermembrane space, and the proton gradient then wants to diffuse back across the inner mitochondrial membrane, but it can only go through the ATP synthase with a proton motive force, which means that a conformation change of the protein happens, forcing ADP together with phosphate to make ATP. At the end of the chain, there is the importance of oxygen finally. So this is where oxygen is used in respiration. And this orange block is just to show you it all coming together. There isn't actually a big orange blob there. The oxygen molecule becomes reduced, accepting hydrogens and electrons to make 2 water molecules. So if you remove that oxygen, the electrons cannot no longer travel through the chain and the whole thing would shut down. FAD is slightly different. FAD attaches to the second cytochrome, and FAD allows its electrons into the second cytochrome, whereas the hydrogens remain in the matrix. Now, from NAD, we get basically 4 hydrogens per electron pair that goes through in electron carrier 1 and 2, and electron carrier 3 per electron pair that comes out. We get 2 hydrogens going through, so we get a total of 10 hydrogen ions because of NAD traveling through to the mitochondrial matrix. For reduced FAD, we get 4 from electron carrier 2 and 2 from electron carrier 1, so we get 6. So in total, there's 16. ATP synthase, then we need to be a bit more familiar with how ATP synthase works. So ATP synthase is has a drum-shaped part located in the membrane. Let me just get my highlighter here to help us out. Okay, so we've got part C, which is a drum-shaped part. Okay, so down here you can see part C, and there's identical subunits with proton binding sites. The rest of the structure has 2 channels. Okay, the first allows protons in intermembrane space to bind to the subunit. So you can see here the alpha structure down here. Okay, which allows the protons in the intermembrane space to bind to the drum subunit, and the second allows the protons to exit to the matrix. Okay, so again, the second will be up the top here. The 2 chemicals are not aligned, so the drum uses energy from protons to rotate before it can be released, and this is called a proton motive force. So basically the energy of the hydrogens moving through causes the ATP synthase to rotate, and that creates the ATP, forces the ATP to be formed. If you look at stalk Y, okay, which is sorry, my pen is not doing what I want it to, which is sort of here. Okay, stalk is attached to parts A and B, and alpha subunits are active sites for phosphorylation of ADP to phosphate. The beta units change shape, and that causes the ADP and the phosphates to bind to make AT. Not just glucose can be respired, so that's important to know. So we mentioned that earlier on. And effectively, the main things that are respired is carbohydrates. So glycolysis is the first stage, so you can do it in anaerobic respiration. Just letting you know, though, the glucose yield of energy is 17 kilojoules per gram. And this is quite low because 50% is oxygen, so it doesn't generate energy. However, if we do fatty acids, the yield is about 37 kilograms joules per gram. So it's a lot higher. If you think about it, it makes sense. There's less oxygen and there's much higher levels of hydrogen. And hydrogen is what is using to rotate the ATP synthase. And just to remind you before we move on, oxygen is essential as the final electron acceptor and hydrogen acceptor. Oxygen is reduced into water, which is a waste product of respiration. If you remove that oxygen, you will cut off the electron transport. Happen then when we remove. As we know, glycolysis is the first stage, and this So we talked about that. Obviously know what happens then to the reduced NADs. How do they lose the hydrogens if the electron transport chain shuts down and oxygen stops? Well, the NADs must be reoxidized somehow, and that is what you need to know for anaerobic respiration. Key thing for anaerobic is very little very little ATP is going to be made. So in terms of anaerobic respiration, there are two main processes. There is lactate fermentation, which happens in mammals, and that is where pyruvate from glycolysis is used to actually re-accept those hydrogens. So the pyruvate is reduced, accepting hydrogens from the NAD using an enzyme called lactate dehydrogenase to turn it back into lactate. And then the oxygens from the oxidized NAD can then be reused in glycolysis over and over again. This unfortunately means that the lactate builds up. Lactate needs to be broken down because it reduces pH and causes muscle fatigue. Hence, you have oxygen debt when you respire anaerobically. Plants, it's very different. So in plants, pyruvate again is produced in glycolysis, but then a different reaction happens using enzyme pyruvate decarboxylase, which removes a carbon dioxide from pyruvate to give us the product ethanol with an A. That then is reduced by giving the hydrogens from reduced NAD, oxidizing the NAD and that turns it into ethanol. And again, ethanol isn't something that the yeast particularly wants, and it will survive it and tolerate it, but over about 15% the yeast will actually be killed by the ethanol. So again, it's a waste product and it's not good for the organisms. But by doing this, the NAD is reoxidized, and that can then go back to pyruvate to accept more hydrogens and be reduced again. You need to be able to summarize the difference between the two, and you need to be able to say the products of the two and understand that the extra stage, the fermentation stage effectively, where you're making either lactate or ethanol and carbon dioxide, is to re-oxidize the NAD so it can go back to glycolysis and be used again. As I said, ethanol and lactate are toxic, so we need to break them down if possible. But ethanol can't actually be broken down, so it's simply excreted, and that's obviously used for us to gain ethanol from yeast or plants. Lactate is reversed back to pyruvate in the liver, and the liver can do a couple of different things. The liver oxidizes 20% of it to carbon dioxide and water using aerobic respiration. When oxygen is available again, the rest can be converted to glycogen for storage for later use in respiration. Oxygen is needed, though, obviously by the hepatocytes to metabolize lactate, and that is why you have oxygen debt.